Okay, so I'm here, as it says on the schedule, to talk to you about epigenetic markers of stress and trauma exposure. My name is Richard Hunter. I'm an associate professor of psychology at the University of Massachusetts, Boston. So I'm a little down the road from some of the other faculty. And my work over the years has focused on stress and trauma and how that affects molecular markers in the brain, how it affects gene expression over the past 10, 15 years or so, how that affects epigenetic markers in the brain and other parts of the body. So what is epigenetics? The term was coined in the 40s by Conrad Waddington. And his goal at the time, and I give you this context because it's important to how we understand epigenetics to this day, is to how to explain how phenotype was influenced during development by interaction between the environment and the genes. So how does an environment have an impact on phenotype, and how does the genome sense the environment so that development can proceed in a way that's adaptive to the environment in which the organism finds itself? Broadly speaking, Waddington's definition of epigenetics was that it's any transmission of information above the level of the DNA coding sequence. And so from that broad perspective, you could say that, if your parents spoke English and you speak English, that you epigenetically acquired English language proficiency, right? And that seems really broad, but as we'll talk about, when you actually get to real life cases of epigenetic transmission where we can pull apart all the details as we can in rodent models, and we'll talk about one case of that just to sort of explain how these things interact. Behavior turns out to be an important means of transmitting these molecular changes, which in turn contribute to changes in behavior in later life. And I'll unpackage that as we go through a little bit. But there is this interaction, between the behavioral and social environment and these molecular markers. So behavior does turn out to be important. Obviously, in mouse models, we can't teach them English, but we can do other things that are relevant to their sort of ecological niche. Generally, though, nowadays we're talking about molecular mechanisms or molecular epigenetics that basically transfer information and store information in ways that affect gene transcription, right? So a lot of the outputs we talk about with epigenetics are changes in gene expression, which change phenotype. Does that all make sense thus far? Okay. It's an increasingly complicated area. I'm not going to cover every aspect of epigenetics now. It would be too much for the time we have. So I'm going to try and give a general introduction to the methods and different epigenetic marks, which are just these modifications of different things like DNA or the packaging proteins around DNA. But it's not going to be comprehensive because that's just impossible in the time that we have. But if there are areas that I don't cover and you're curious about, I'll do my best to answer any questions about methods or approaches that you have, some curiosity about. Okay, so to contrast epigenetics to classical genetics, one of the important things that epigenetics has shown us over the past 10, 20 years is that our genome is able to dynamically alter its behavior in response to environmental inputs. And that's a big shift from, the consensus about what genes did and what genomes did in the 20th century, which was that genes were relatively fixed, and once development had finished happening, that basically their expression and the phenotype of the organism was not going to change over much. And that, the idea was that development proceeded in a clockwork fashion and wasn't responsive to the environment. We now know that's not true. Again, the important thing about epigenetics is it's not Lamarckian in the true sense, is that you're not changing any underlying genetic code. But interestingly, in some cases, these epigenetic effects can be inherited, and I'll talk about sort of the best described case in rodents shortly. And importantly, for sort of the subject matter of this workshop, epigenetics can help explain why similar individuals can be changed in lasting ways by their environment, particularly by stress and trauma, so that they wind up on different trajectories in terms of the behavior, in terms of pathology, and things like that. So this is probably not new to all of you, but to make sure that we're adequately distinguishing between genes and epigenes. A gene sort of classically is a segment of DNA that codes for a single protein. We now know that's a little more complicated than that, but that's sort of a good basis for us to start about. And again, referring back to the sort of 20th century consensus about how genes worked and how they produce phenotype was Crick's tongue-in-cheek central dogma of molecular biology, which is DNA codes for RNA, which in turn codes for protein, and protein is the thing that gives you a phenotype as a cell, and then the sort of composite phenotype of cells in the tissue give you a tissue structure, and so forth and so on until you have a whole person or rat, whichever you may be. And this is a little more involved now in terms of how this kind of stuff can happen, but this gives you an idea about some of the areas at which epigenetic mechanisms can operate on this sort of linear classical transmission of information and change it so that you can change transcriptional control here, which is a major place where epigenetics is understood to happen. You can change how RNA itself is processed, how RNA is transported and localized within the cell, how it's degraded, how it's translated into protein, how those proteins in turn are modified. So in this case, we'll talk about histone modifications. These are the packaging proteins for DNA. They're actively modified, and that affects transcriptional control. So there's interactions at all of these levels. And in fact, if I was to give you more than a cartoon of this, there would be a lot more lines. It would be and a lot more complex. But this is just here to give the point that this sort of classical genetic flow of information is modified and interacted with at pretty much every level by various epigenetic mechanisms. And so that'll be important as we go on. And another aspect of this too is that in terms of RNA processing control, there's another emergent field parallel to epigenetics called Epi Transcriptomics, where RNA itself is modified in the same kinds of ways that DNA is, and that in turn affects how it's processed, how it's localized, and how it's translated into proteins, et cetera. Okay, so what is molecular epigenetics? It is basically the modification of various molecules that interact with DNA in some way or another. It can be the DNA itself, as you see here, in the case with DNA methylation. We now know that DNA is modified in at least 4 or 5 different ways. This cytosine methylation is the classical way in which that happens, but there's also hydroxymethylation, which happens actively in the brain. And now we know there's adenosine methylation is also known to happen in the brain and in other tissues. Then there are these modifications of the histones, and roughly every 140 bases of DNA is wrapped around what's called a nucleosome, which is an octamer or 8-piece ball of these histone proteins. And they have tails that stick off of them that can be modified in various ways. So they can have acetyl groups put on them here, or they can have phosphate groups put on, they can have methyl groups. And these modify the local structure of the DNA. So the acetyl groups, because they're large and charged, tend to push the DNA apart and make it more accessible. So highly acetylated regions of histones tend to be associated with highly transcribed genes. Methylation, on the other hand, is a pretty hydrophobic group. So it doesn't like the watery environment inside cells, and so it tends to wrap the DNA more tightly. And so the regions of the genome that have high levels of DNA methylation and of histone methylation tend to be much more compacted, and they form what's called heterochromatin, which is silence genes or silent sections of the genome. And heterochromatin, as it turns out, is really important for determining developmental cell fate. So you, in a certain sense, you can just look at where the heterochromatin is in the cell, and that can tell you what kind of cell it is, because what you've turned off predicts a lot about what your phenotype is. So this is sort of where, Waddington's insight about developmental interaction between the environment and the epigenome was sort of so prescient, because it turns out that the molecular mechanisms by which we develop tissues like brains or behaviors are very much conditioned on these molecular epigenetic mechanisms. And then the last category, which is the largest and the most complex, are non-coding RNAs. And I've just listed a few of the many types of non-coding RNAs here. The most common ones that are studied now in humans context and clinical context are microRNAs, which tend to be involved in affecting protein processing and degradation, and long non-coding RNAs, which have a whole variety of functions. We wrote a review on this 2 years ago now, where we outlined at least 18 different functions that long non-coding RNAs can have. That list is expanded since then. So this is a pretty large category of epigenetic mechanisms that we're only really starting to understand. And in fact, a lot of the work that I do is focused on discovery of new non-coding RNAs, because it turns out that our genome is full of these things. We long thought that the protein coding genome was the only thing, that the genes that coded for proteins were the sole determinants of phenotype and behavior. But in humans, that's only about 2% of your actual genome in terms of code of sequence. And it turns out that about 50% of your genome is comprised of transposable elements. These produce RNAs. Many of them are turning out to be non-coding RNAs that have regulatory functions. So we already have identified somewhere near 80 to 100,000 non-coding RNA genes. So this is a big exception to Crick's central dogma that, DNA codes RNA, codes protein. Sometimes it just goes DNA to RNA, and then the RNA does something much like a protein. And 100,000 long non-coding RNAs is 5 times the number of genes that are present in the protein coding genome. So the complexity of the genome and its capacity for self-regulation is approaching an order of magnitude greater than what we thought at the turn of the century. And that continues to change because it's possible that as many as several 100,000 of these genes exist in our genomes. Okay, so is this sort of general overview clear thus far? Okay, cool. Yeah, and please do stop me if you have any questions or if you just find yourself getting confused, because it is on me to explain this stuff. That's why I'm here today. Okay, so DNA methylation, hydroxymethylation. This is probably the most important one if you're interested in clinical populations, if you're looking at stress in humans. It is most commonly studied, and part of the reason for that is that DNA is the most stable of the molecules we're talking about here. I'm sure you've heard cases about the Neanderthal or Denisovian genomes that have been extracted from hundreds of thousands of year old bones lately. That's because DNA is a very stable molecule and sticks around easily. So you can get blood swabs from people in field environments, and that DNA will stick around. RNA degrades pretty quickly, and protein also degrades pretty quickly. If you've ever left a steak out, you know this. So DNA methylation is often the easiest of the epigenetic modifications to study in human populations or in the field if you're a field biologist. Histone and chromosome modifications changes these proteins that package DNA, and so change the accessibility of DNA to the machinery that transcribes it and produces phenotype. Non-coding RNA are these huge variety of RNAs that perform various regulatory functions throughout the cell. So, and again, to emphasize this, most of what we're talking about when we're talking about molecular epigenetic changes are changes in gene expression. So these two things are pretty tightly linked. So gene expression is sort of the thing that drives phenotype, in whether it's in neurons or blood cells or whatnot. Okay. So as I'd mentioned, this is really important in development, like sort of the pattern of the epigenetic landscape, as it's called, determines cell fate. And it also tells us something important about epigenetics that makes it a little more challenging to work with than genetics. Every single cell in your body, with the exception of your gametes and your gonads, has the same genome. That is not true of the epigenome. The epigenome determines or allows us to have neurons and skin cells and kidney cells from the same genome. So each set of epigenetic marks, each landscape is specific to the tissue and even to the individual cell that it's in. And this is an important issue when you're thinking through study designs is, a lot of stuff that we do in humans who are exposed to stress is in peripheral blood. And that makes it challenging when using epigenetic measures to go from something like peripheral blood or saliva, which is mainly epithelial cells from the inside of your mouth, and extend that to assumptions about what's going on in those people's brains. And in fact, I would say that's probably the biggest technical challenge right now in terms of epigenetics, and it has been for years, is the tissue specificity of the marks. And I'll talk a little bit more about that later, but it is a challenge, and it is something to keep in mind whenever you're doing an epigenetic analysis, that you can't assume that because you see something in white blood cells or in epithelial cells that the same sort of environment or same set of changes holds in the brain. Okay, so the reason I'm interested in epigenetics is because I'm interested in stress, as everybody here is. Otherwise, you wouldn't have paid for the workshop. And one of the ways I conceptualize stress is a challenge that an organism has to adapt to, and it's one of these places where epigenetics necessarily must play a role. If you want to if you're exposed to a traumatizing environment or something that's dangerous, and you successfully adapt to it, you probably are going to change your behavior around that. And so the question had long been in stress is why does stress change behavior? Why does stress change all of these physiological markers over time? And how does that stay encoded? How does it get under the skin, in the words of Bruce McEwen? And so epigenetics should play a role here as a mechanism. And the first proof of principle of this sort of environmental influence on epigenetics was in rats. And I'm going to go through this. I know a lot of folks here are interested in the clinical applications of this, but I think this is a useful model because we understand it in a lot of detail, at a level of detail that we can't sort of examine in humans for some of the reasons I've listed about tissue specificity, et cetera. And so Michael Meany, Paul Plosky, and others in the late 20th century demonstrated that there were patterns of behavior that were transmitted between multiple generations of rats. And that this happened without a change in the DNA sequence. And so this was the first evidence that there might be a molecular basis for epigenetic transmission in mammals, at least. And what Meany and his colleagues did, specifically in Weaver, was they did a whole series of experiments that were really beautifully conceived, and they sort of phenotyped these animals, and they discovered that within a normal lab rat population or lab mouse population, there are variations in maternal behavior. And so rats tend to groom their offspring by licking them, or perform archback nursing, do things like retrieve pups if the pups fall off the nest. These are all sort of classical rat mom behaviors. And some rat moms do a lot of that stuff, some do very little. And it turned out that the ones that do a lot had daughters and granddaughters who do a lot. Those who do a little have daughters and granddaughters who do a little. And that kind of variation isn't really surprising. You'd expect some kind of variation in behavior in any population you look at, just because statistics. But what was interesting about it is if you cross-fostered these animals, like if you actually took fetuses out of one mother and put it into the other mother, those babies would grow up to have their birth mother's phenotype, not their biological mother's phenotype. So it wasn't a genetically transmissible effect. It was a parenting transmissible effect. It had something to do with the maternal environment. And in fact, through a lot more detailed experiments, they found that basically by using wet paintbrushes to simulate licking and grooming, they could change the behavioral phenotype of the low licking and grooming babies so that they showed up as high licking and grooming animals for the rest of their lives. And that was in turn would be transmitted to their offspring, to their daughters and granddaughters. So they showed that it was the licking and grooming behavior that was important. And then in a study done led by Ian Weaver in 2004, they showed that glucocorticoid receptor levels in the hippocampus, and glucocorticoids are the classical stress hormones. I'm sure you've heard plenty about them at this point in the workshop. The receptors for these things were reduced in the low licking and grooming animals and increased in the high licking and grooming animals. And in turn, these animals showed differential response to stress. The low licking and grooming animals were more stress reactive. They were more anxious when you looked at them in the lab. And the high licking and grooming animals were less anxious, less stress reactive. And that's thought because there's good glucocorticoid receptor feedback there. And beyond this, they also discovered that in these animals, there was an increase in methylation of the DNA, specifically in what's called the 1,7 promoter region of the glucocorticoid receptor. And so that DNA methylation was suppressing transcription of glucocorticoid receptors, and that was the actual molecular mechanism by which this behavioral transmission happened. It's a really surprising set of findings that methylation in one brain region altered behavior in such a substantial way, and also that behavior was changing gene expression in such a persistent way that it could be transmitted across generations. And so there, again, the reason I'm bringing this up is because it's this behavioral element interacting with the molecular aspect that's really important. A lot of cases where we look at epigenetics, we focus very much on these biological outcomes. We focus on the molecular epigenetics, but the importance of epigenetics is these environmental interactions. And so understanding that they almost always are happening in interaction with the environment, and that the environment is a really important variable is one thing that like to transmit to you today, if you'll pardon that. Okay. And it turns out that it was even more complicated than this when you get down to the details. So these high-licking grooming animals with lots of glucocorticoid receptor expression had better feedback with their glucocorticoids. The GR gene was during development while the mother was nursing and grooming their offspring, that was affecting serotonin levels in the babies. That in turn was affecting how a certain transcription factor, NGFIA, was interacting with the GR gene, and that in turn was deciding or helping the methylation machinery decide whether to methylate and turn off this gene or turn down this gene, or whether to turn it up. And again, so this is sort of one of the most complete stories in epigenetics. So I'd sort of give it to you as an illustration. We don't have this deep detail of understanding about humans, although there have been studies since then looking at how the methylation of the equivalent part of the glucocorticoid receptor gene, the 1F promoter, are changed in humans with different histories of trauma and childhood adversity, et cetera. Some examples: there are differences in DNA methylation at this promoter in suicides, as well as some other stress-related genes. There's somewhat controversial literature on people who self-report childhood abuse showing similar patterns of methylation. There were some studies that have validated that finding, some studies have not. Whether that's an effect of self-report studies or not is hard to say. As I'll show you in a second, people with PTSD and a history of abuse show broad genome-wide changes in DNA methylation. And then people exposed to extreme trauma often transmit. Oh, Katusis, do you have a question?
Yeah, in these particular cases, what was studied? I assume that's just blood was studied, or was it?
Yeah, it's usually blood or saliva. So there haven't been many cases where brain tissue, in the case of suicides, they have been able to look at some brain tissue and look at epigenetic changes. But the vast majority of cases where you're looking at clinical populations, for obvious reasons, you can't dig into their brains.
Okay.
So most of this is peripheral stuff. Yeah. And I would say that the conceit there, like sort of the assumption that you like to make when you're doing this stuff, is that given that stress has a humoral effect, the hormones are circulating throughout the whole body, you would expect changes in stress sensitive genes in the blood just as much as you would in brain. And so we make that kind of conceptual leap that, well, if we see changes in how stress related genes in blood are being regulated by epigenetic marks, probably something similar is happening in brain. And we do have enough data from animal studies and from postmortem tissue now to assume that, at least in case of the glucocorticoid receptor, that there does seem to be some correspondence between what you see in peripheral blood and what you would see in at least stress-sensitive brain regions like the hippocampus or prefrontal cortex. But yeah, very important question. So we also know too that people who have been exposed to extreme trauma do transmit some kind of epigenetic information to their offspring. The most famous cases of this are early cases of this are some of the work that Rachel Yehuda at Mount Sinai has done with Holocaust survivors, but more recently, people like Elizabeth Binder and Yehuda have done with folks who went through the 9/11 disaster in 2001 or Hurricane Katrina in New Orleans. And it seems like there's transmissible aspects of this that go on to their children. In the case of Holocaust survivors, their children are much more likely than relatives whose parents did not go through the Holocaust to wind up with PTSD as adults. They also show changes, as I'll show you in a bit, in methylation of the glucocorticoid receptor. So again, the glucocorticoid receptor is a regular focus of this kind of stuff, but I do want to emphasize that it's not the only thing mediating this stuff in humans. It just happens to be where we have the most evidence that epigenetic effects are happening as a consequence of trauma and in terms of transgenerational transmission of that trauma. Okay, so Katusis, you already sort of got ahead of me on this question. So you have to be cautious about particularly human studies when you're thinking about what tissue you're using. You can't assume, although we do, that blood is going to have the same kind of epigenetic landscape as the brain does. But again, if you focus your attention on those things that are stress responsive, those genes that are stress responsive, as opposed to those genes that sort of determine what a white blood cell is versus a neuron is, you're more likely to get stuff that's in concordance. So Another sort of aspect of the context is that sometimes we look at these global changes in methylation or histone modifications. Usually the practice is to look at specific sites. So I've talked a lot about the glucocorticoid receptor. So looking at specific parts of stress hormone receptors is more common. And there are a couple reasons for that. One is that it gives you a gene readout to target and look at in other assays, so you can kind of do some correlative work and demonstrate that the effects of the modification you're looking at are real. And the other is it's just cheaper. Sequencing is pretty expensive, even though it gets cheaper, month over month. Looking at specific targets is a lot more tractable for most labs to do. So epigenetics is operating through these molecular interactions in the cell, and this is changing this information flow from the genome to the rest of the cell. We've talked about DNA methylation, and we'll start to get in now, like how do you actually look at this? DNA methylation, in contrast to most of the other modifications we're going to talk about, is relatively stable. So it can persist over years to decades in many cases. But an important caveat about all of these things is that even these sort of stable modifications it's not like, you scratch an engraving into a piece of stone and it stays there. Most of the epigenetic modifications are maintained because there's a stable interaction between what are called readers and writers. So there are things that put the mark down, and then there are things that remove it. And so this is sort of like more like a standing wave than it is like a standing stone. All of these things are dynamic and can be perturbed by changes in the balance between the machines that make the marks and the machines that take the marks away. Okay, so histone modifications we've talked about a little bit. The most commonly examined marks are methylation and acetylation. There's a little bit that's been done with some other marks like phosphorylation, proteinylation has seen some, and there's been some very interesting papers recently showing serotonylation and dopamylation. So actually tacking on these neurotransmitters to histones has been shown to have behavioral effects, at least in a few animal studies thus far. We'll see if anything comes out in terms of the human literature. So a little more about what we're talking about with histone modifications. This is from an old David Alice paper. But you see here, you have these 4 canonical histones, H3, H4, H2, A and B. There are 2 of each in this histone octamer, in this nucleosome. They all have little tails that stick off. And there's a nomenclature here where you have the tail, and then you have the numbering of the amino acids in that tail and the modifications that happen there. So for instance, you would see, if you're looking at this literature, H3K4 can be acetylated, it can be monomethylated, it can be di and trimethylated, and each of those modifications of this particular histone indicates something different about the state of that local environment in terms of gene expression. So typically, say, if you have this particular residue trimethylated, that's usually associated with promoters of actively transcribed genes. In contrast, this sort of next residue down, the 9 lysine, when it's trimethylated, you're usually looking at a heterochromatin or a silent section of the genome. So each of these sort of operates as a molecular switch, determining to a certain extent, the chromatin state of the local environment, and therefore what the genes in that environment are doing. And sort of in a very gross sense, euchromatin, which is the active part of the genome in an individual cell, is associated with a lot of acetylation, fairly low amounts of methylation, and heterochromatin, which is the silent structures that help determine cell fate, is associated with lots of methylation, relatively little acetylation, for the sort of chemical reasons I was talking about before. And there are particular marks, K9 and K27, that tend to be associated with, when trimethylated, with heterochromatin. When they're acetylated, they're more associated with more active chromatin regions, etc. Is that all relatively clear thus far? I know it's a lot of acronyms and things like that, but the take home is that generally speaking, methylation is associated with suppressed chromatin, with the exception of K4 methylation. And generally speaking, acetylation is actively transcribed genes. Non-coding RNA, I've already sort of mentioned some of this. It represents most of what the genome actively transcribes. So 70% of what's transcribed in an individual cell's genome is non-coding RNA versus about 5% from genes. Non-coding RNA is capable of regulating cell functions at a variety of levels, as I've mentioned. It has a bunch of different flavors, and new ones are getting discovered all the time. As I say, there are at least 18 to 20 different modes of action for these non-coding RNAs. MicroRNA, again, is the one that you'll see most often in the literature for clinical populations, and I'll talk about a little bit about that as we go through. Okay, and this was just from a paper we wrote a couple of years ago showing the variety of different things that non-coding RNAs can do. MicroRNAs, their main mechanism of action is to bind to other target RNAs or to proteins and cause their degradation, and they do that through interacting with proteins called DROSHA and DICER. I'll not get into a lot of detail about that unless you're curious about it. But they basically, one of their major functions is to suppress the expression of viral RNAs in the cell. So they're part of the cellular immune system, but they also have these regulatory functions in terms of determining which genes transcribe RNAs and which RNAs actually become proteins. So they're important for the balance of protein expression, RNA expression in the cell. We know that some long non-coding RNAs can act as activators of transcription of target genes. Some of them can repress them. Some can enhance it to some extent or another, so help move transcription factors to targets. Very often they're involved in determining the chromatin architecture. So we know a lot of how the genome decides where to put heterochromatin is dependent on the RNA that's expressed at low levels by the target tissues or the target parts of the genome for that heterochromatization, so that they can form a scaffold for the heterochromatin. And then a number of other different functions are possible for non-coding RNAs, all of which can affect the information flow in the cell and then in turn the phenotype of the cell and by extension our behavior and our physiology. Okay, so is that all relatively clear thus far? Any big ambiguities?
Is it okay if I ask you a tricky question? It just crossed my mind. So if these modifications are tissue specific, and whatever is in the brain is not necessarily reflected in, other tissues,
right?
How is that inherited then?
So yeah, I think this is the question, and I think that's one of the reasons I sort of led with the rat study, is that in rats at least the heritability is via the behavior of the parent. So the parent's behavior has changed persistently because of the change in glucocorticoid receptor expression and the epigenetic regulation of that in their brains, but the transmission is the behavior, right? And so we think the same thing is the case, say, with Holocaust survivors and the like, is that they have a behavioral phenotype, which is probably hypervigilant, is probably very, very PTSD-like, because you imagine that population of people probably all have PTSD. And that is probably the mechanism of transmission to the offspring. However, all of that said, there is evidence now that there is germline transmission of some of these non-coding RNAs, both through sperm from fathers, which is a little rarer case, but does seem to happen, and in the eggs from mothers. So there's definitely behavioral transmission, and I suspect that's probably the more common means by which these things become persistently embedded in the brains of people's children if they have trauma exposures. But there is also evidence that some of the non-coding RNAs that get packaged into eggs and sperm do transmit these liabilities as well. But to date, we haven't really been able to show that in humans. That's really exclusive to animal models, which I would say should always be taken with a certain amount of caution when you're thinking about human stuff, particularly in sort of the neuropsychiatric disease area, because we haven't had a really good history of translating that stuff. Does that answer the question?
Yes, and I'll just take another minute. I was thinking about two examples. Well, let's say birds, when they see the hawk, even very little, well, just newborns, right? Mm-hmm. They hide in the nest, right? Mm-hmm. Or humans, whenever we see a snake, we just shy away from it. We jump to the side, right? Mm-hmm. If these are behavioral phenotypes, right?
Yep.
Somehow we know that the snake is dangerous, even though we've never seen our parents to jump away from a snake, right? Right. So it becomes really interesting, to see what is the inherited pattern. I don't think it's coded into the DNA.
Yeah, it's an open question, but we do know with phobias that, for instance, that people are more likely to be phobic against things like snakes that were dangerous to us in our sort of evolved environment as human beings. Something like in some cultures that are still living, in more primitive conditions, where snakes can account for something like 20 to 25% of all mortality. And so it's thought that, in our evolved environment, we built some kind of something into our DNA or into our epigenome that made us afraid of snakes sort of fairly naturally. So it's easy to get phobic to snakes because snakes really are dangerous if you're a human. But I don't think anyone has nailed out exactly what genes or epigenes are responsible for that. But I do think it sort of speaks to this idea of transmissibility of fear and things like that. Angeli?
Quick question. So this makes me think of two things. It sounds a lot like AI learning, like that process by which the computer kind of tries different things and gets rid of things. And there's a point where humans don't know what the computer is learning.
Right.
It sounds a lot like that. And the other thing that I was thinking was a lot about the epigenetics is how females are born with a set number of ovules, and they live with females throughout their lifetime. And so the ovules are exposed to everything that the female is exposed to. And then that in turn is transmitted to the child in some way. Has there been any relationship or anything relating? I know that maternal RNA is a big issue that it can be tracked very linearly back generations and generations. So has there been any study looking at more of that, the importance of epigenetics within the maternal RNA, maybe even the mitochondrial RNA that is transmitted, and so on and so forth? I'm having kind of like, sorry.
No, don't apologize. They're all good questions. And excuse me, mitochondrial transmission is a thing and contributes to a lot of different disorders. It is mainly matrilineal. I don't know if there have been epigenetic changes demonstrated in mitochondrial genes that are transmissible. We had a paper 2 years ago where we showed that people with a history of trauma and experience of racism had increased methylation on a particular mitochondrial gene that seems very responsive to stress hormone receptors. So there is a little bit of evidence that mitochondrial, and there's also evidence that sort of mitochondrial quality can be affected by stress. So that's potentially an issue there. And there is some thinking that that's part of the reason that you sort of stop making oocytes later in life, because that is under epigenetic control. It's not inevitable that you stop making oocytes. There is a organismal level decision to stop doing it at a certain time. And the thinking is part of the reason for that is quality control for the mitochondria, because mitochondria, one of the things that epigenetics does is protect genes from damage. And mitochondria, because they're basically bacteria that invaded our cells billions of years ago, don't have those protective things. They don't have the histones that protect them from oxidative damage. And there's a place where you make all the oxidative stress, reactive oxygen species. So mitochondria tend to accumulate a lot of deficits in their genomes over time. And so one theory, at least, of why we run out of oocytes after a certain age is because the mitochondrial quality declines so much that it sort of risks defects in the offspring. But there is actually evidence that you can turn oocyte production back on by playing with some of this epigenetic machinery. So yeah, it is an interesting question. To my knowledge, I don't think anyone has actually shown in humans that there's changes in mitochondrial gene expression. There are definitely studies, Tracy Bale and others have done, showing in rodent models that there definitely is maternal RNA alterations by stress history in the mothers and things like that does get passed on to the offspring. But it's still relatively early days, and I think that's only really been looked at for the past 4 or 5 years in any detail. But I think it's tremendously important, and it really is, again, as you say, because like your mother is carrying around these oocytes for her whole life, and her history is being encoded in those to some extent epigenetically, and the challenges she's been through and things like that. So there probably is some fairly powerful transmission there. That said, it seems like that is weaker than gestational effects or behavioral effects, and I say that just based on sort of the Holocaust survivor stuff and on stuff from Dutch hunger winter, some of the stuff from the Winter War in Finland with children who were born when their mothers were starving. Those have very powerful effects, whereas the children born on either side to the same mothers wind up relatively normal, which suggests that sort of like that maternal environment right at that moment while mother is going through something traumatic is more significant than her total life history of trauma. So I think I can't answer your question completely, but hopefully that sort of gives you a little more context. And also, I think the AI question is really interesting because it's like nowadays we use AI to look at epigenome or epigenetic landscapes and things like that. So and a lot of the folks I know who do that kind of stuff in cancer biology, because cancer biology is a lot further ahead of neuroscience on this sort of stuff than most neuroscientists would like to admit. They're very concerned about it because it's like, are we really doing an experiment when we don't really know what the experimenter is doing and we don't know how it's deriving rules and things like that? And yeah, there is a level of complexity here that is really intellectually challenging, but also fascinating. So I hope that is an adequate partial answer to your questions. Any other questions or thoughts? Okay, I'll keep on moving. Okay, so how do we measure these things? A lot of things, as I mentioned, you look at single targets. Very popular ones are obviously things that are involved in the stress axis as normal function. So 11 beta HSD, glucocorticoid receptor, FKBP5 has been shown both genetically and epigenetically to be, and this is one of the packaging chaperone proteins for glucocorticoid receptor that determines its sensitivity. Epigenetic changes or genetic changes in it determine risk of PTSD after trauma. Sort of the classical monoamine related genes like serotonin transporter, CMT, dopamine beta hydroxylase, BDNF as sort of the major neuroplasticity gene, or at least the one that's best studied at this point. There are definitely methylation effects on it in response to stress. And then a bunch of neuropeptides, oxytocin, CRF, and AVP are sort of the obvious ones as they relate to stress axis function. So these are sort of the common single targets if you want to do single level analysis. Another area where targets are of interest now, particularly given the sort of neuroimmune interactions and the neuroimmune hypothesis of things like depression, are also pro-inflammatory cytokine genes and sort of master regulators of inflammation like NF-kappa beta, et cetera. So these are targeted analyses that one can do. And when you're thinking about targets, obviously it should be something that's related to the question you're interested in. If you're interested in inflammation, then looking at glucocorticoid receptor as an anti-inflammatory target would be useful and interesting. Looking at NF-kappa beta as a pro-inflammatory target would also be useful and interesting. It's easier to work with things that other people have validated before, in part because you can nowadays often buy the assays for those things off the shelf as opposed to having to design your own. So that's a methodological concern. We don't have a clear global picture, so one of the problems with the approach of looking at, and to use my favorite scientific metaphor, the drunk underneath the lamppost, where he's looking for his keys and he hasn't found his keys. You ask him why, it's like, well, I don't know. Like, well, why don't you look somewhere else? Well, all the light is here. So we have a bit of a bias in this field, as in many areas of biology, to keep looking where we've already found things. I don't think we're done with figuring out all the things that are epigenetically involved in stress responses. But we are getting there because of more global approaches, and I'll talk about those in a second. Okay, so the one big thing that has changed in terms of genetics and epigenetics in the past 20 years is the introduction of next generation sequencing. And this covers a whole slew of technologies that I don't have time to talk about because that's a whole lecture in unto itself. But basically, previous techniques, like the ones developed by Sanger, could only sequence about a few hundred bases at a time. So you could get 300 bases, but you have a genome that's 3.2 billion bases. So needless to say that it was really challenging at best to be able to construct a whole genome sequence, much less be able to sort of look at every RNA expressed and then a bunch of individual cells. But NGS basically allows you to sequence trillions of bases in a single run that takes a day or so, which is the equivalent of hundreds of whole human genomes on a single instrument. So we have just orders and orders of magnitude more capacity to look at these things now than we used to. And in addition to this, we've sort of bootstrapped this technology into a way at looking at epigenetic change. So we can look at parts of the genome, as I'll talk about in a little bit, that can be modified by these epigenetic marks. Okay, so typically these approaches, you're extracting and fragmenting some nucleic acids. They're then sequenced as reads. Typically, they're somewhere between 50 and 2,000 to 5,000 bases, depending on the instrument using. Most common instruments are running 100, 200 base reads. Then you have to go through a fairly computationally complex task of aligning to a reference genome. If you're working in humans, this is pretty straightforward. Very often that alignment happens by the core that's sequencing your stuff. So you'll have these things aligned for you. And then depending on what you want to do, you can analyze those in a number of different ways. A very common one is just looking at differential gene expression. How much more RNA for this gene do we have under a stress condition versus under control conditions, et cetera. Okay, so you can also look at DNA and RNA simultaneously. So you can look at the underlying DNA, you can look at the epigenetic landscape underlying it. So very often people will look at this whole epigenetic landscape for a particular set of marks. So they might look at histone methylation, in a particular cell type or in a particular tissue, and then look at the expression of genes in that tissue so that they can correlate the gene expression with the epigenetic changes. And in fact, nowadays, in the past year or so, it's become possible to look at the epigenetic landscape in a single cell from a single piece of your brain or a single blood cell, and also look at the RNA or the gene expression that it has as well. So we've gotten to a very fine scale of analysis that's possible nowadays. But of course, as you might guess, all of this data creates a lot of sort of analytical problems for us. Okay, so it's pretty complicated. The preparation and fragmentation requires some expertise and optimization to do it. You need specialized equipment. The sequencers are $500,000+ So getting into this, although you can pay someone else to do it less and less, you can do these projects for a few $1,000s now as opposed to tens or hundreds of thousands sometimes, still requires the infrastructure to do this. So you usually are going to be working in association with a core at your university or with a private vendor or something like this. This is doing this stuff yourself is very challenging unless you happen to have $10 million, in which case we should talk after class. That was a joke. Okay, so how do we measure DNA modifications? So the major ones that folks are looking at now are methylcytosine, which is the best established of all the DNA modifications, hydroxymethylcytosine, and more recently 6-methyladenosine. Most of the analyses are focused on methyl C. It's been around the longest. It's the easiest to work with chemically and otherwise. Bisulfite is the major means of doing this, and bisulfite is basically a means of converting a methylated cytosine into a uracil so that you can read it as a different base. So you can see the change before and after bisulfite treatment and be able to identify the methylated bases that way. You can also approach this through methods called methyl dip, which basically you have antibodies against the particular modification, and you can use those antibodies to pull down that bit of DNA. And then lastly, we have long-read sequencing like that produced by Oxford Nanopore or Pacific Bio, where you're basically running thousands of base pairs of DNA through a tiny nanopore that detects the small electrical differences between the different bases and can give you really fine scale detection of which of those bases are modified and how. And that's probably the best method right now, but it is as you might guess, also an expensive one. So one of the things that's been done with this is looking at sort of whole genome approaches. One of the besides the next generation sequencing approaches, there are also methyl chips. At the time this particular paper we're looking at right now was published, queried about 400,000 different places in the genome. Now it's up to 800,000 or a million, depending on the company and the particular chip you're using. But what we're looking at here is changes in CpGs, and CpGs are basically methylated cytosines that are next to guanosines. These are sort of the most common form of DNA modification, is this sort of particular pair, base pair. And you can see that there's major changes in the methylation status of CpGs in folks who have PTSD in this particular study versus controls. And I'll show you a little bit more data from this one in a little bit, because it's interesting in terms of not only the landscape within people with PTSD, but sort of subdividing PTSD via epigenotype into two just biologically distinct entities, which I think is a contribution that really hasn't been trumpeted enough. And I think it's pretty important as we think about these disorders. But you can see here basically, and what all of this is just the number of sites that they found that were different. And then the different colors indicate the location within the genome. So if it's red, it's within the gene body. If it's blue, it's within the first exon part of it. And then these are for sort of more distant elements of the genome, or so non-coding areas of the genome. Okay, so doing this bisulfite comparison is the classical way to do this. This is particularly easy if you have a specific target you want to look at. Bisulfite sequencing is on that small scale is pretty easy and relatively inexpensive compared to most of the other approaches. You can do the global sequencing, but again, that is a little more involved. The things you can run into challenge-wise with this is incomplete conversion. So maybe all your unmodified cytosine doesn't convert as well. You also have to deal with the confound of hydroxymethyl cytosine, which does slightly different things. And of course, with all of these things, degradation of your sample can be a problem. Okay. Another approach to this is immunoprecipitating, which basically means using antibodies to pull it out of solution. So the only thing, the thing that you're interested in, only that gets pulled down. So you're enriching your sample for whatever modification you're interested in. A more common means of doing this is called CHIP, which is chromatin immunoprecipitation. One of the issues with all the antibody-based approaches is that antibodies are sticky. They have non-specific interactions. You get higher background with these approaches for that reason. Proteins like to stick to proteins. The other issue is that antibodies, because they're so much larger than DNA bases, can't provide you with single base resolution. And this is a problem that shows up in papers from people who know better, is they ascribe a single base resolution to a method that can't really give you more than, like, say 40, 50 base resolution. So these methods that's a big caveat with these when you're talking about single modifications. So using proper controls, comparing to unprecipitated inputs, et cetera, are really important things when you're using these immunoprecipitation methods. Long read sequencing is another way to get these kind of things done. It also is being used as an adjunct to the sort of classical short read sequencing, and by short read I mean that sort of 150 to 200 base sequencing that Illumina does. Illumina is the major producer of short read sequencers, and this allows you not only to look at epigenetic marks, but to get a larger section of DNA with which to, or RNA is the case, with which to identify what exactly is changing in your tissue. And as you can imagine, the longer a piece of text you have, the easier it is to find the exact place it belongs in the genome. And this is particularly true of non-coding genes, because a lot of them contain repetitive element or transposable element DNA and RNA, which means that they can be hard to align to a genome. And like old school approaches, you'd have 20% of your sample you couldn't align uniquely, but using these, you get better unique alignments. And again, stop me if I'm talking complete gobbledygook. Okay, so an example of a single target DNA methylation analysis, this is work from Rachel Yehuda. It's now a few years old. And this is looking at peripheral blood monocytes in parents who have PTSD. And you can see here this significant difference in DNA methylation of this GR1F promoter, which is the analog of the RAT promoter we talked about early in the talk, between parents who both have PTSD and father-only PTSD and mother-only PTSD. And so this tells us a little bit about this, and that basically it seems that maternal PTSD is a lot more significant factor in changing the expression of glucocorticoid receptor, at least in peripheral blood, than paternal PTSD. You can get at this with Sanger sequencing. You can also get at this with PCR-based methods. Most molecular biology labs at most institutions have PCR machines that you can do bisulfite sequencing at this kind of single target level. So this can often be an easy way to get into this stuff and look at single modifications. As you can see, though, humans are pretty noisy compared to rats, so they're pretty wide margins of error here. Also pretty low end in this particular study, but it has replicated in larger samples since then. Okay, so whole genome analyses are less common due to the cost, but they probably should be happening more. One way to get around the cost and sort of get a broader sample is what's called reduced representation bisulfite sequencing. So that's only looking at targets. The same kind of method applies with these alumina methyl chips, where you're only looking at 800,000 or a million or so known CpG islands. On top of this, again, this tissue specificity issue comes in. If you're looking at a piece of brain, even a small piece of brain, it's heterogeneous because it has both glia and neurons in it. Each of the neurons has a slightly different phenotype. So, which is one of the pushes towards doing more single cell sequencing nowadays, so that you can do these kind of differences between individual cells. So that's one of the things that can complicate this stuff, or at least contribute noise to your analyses. And this was what I was referring to before. And this is basically within a sample of PTSD patients. And what you're looking at are basically here are PTSD cases with child abuse history, and PTSD cases without. And you see in both of these cases that basically it's a completely different pattern of methylation and gene expression between these two different subpopulations. And we've known for a long time that PTSD, the history of child abuse, contributes to your liability to getting PTSD as an adult. But this is the first demonstration that basically the two types of PTSD are biologically distinct. And I think this is one of the best cases for the power of these kind of genome-wide, even if they're sort of sampling genome-wide approaches to epigenetic changes in pathologies like PTSD or in stress-induced change, is that you get to see the forest and not just the tree, and which is kind of the downside of the targeted approaches, is that you might miss something that's really striking when you look at this analysis. Like this pattern here with no child abuse is completely distinct from the one with child abuse. If we were just looking at, say, GR methylation patterns, we might sort of have no signal or an inadequate signal. But if you look in this broad way, you see that there's a really distinct change in the overall epigenetic landscape as a consequence of this developmental trauma versus those who don't have developmental trauma and were exposed to trauma only as adults. So this, I think, is an argument for more of this kind of stuff if you can manage to pull it off. Okay, histone modifications can be measured, globally using Western blots and a lot of other sort of basic molecular biology lab techniques. ELISA, and RAA can also be used. If you want to get information about specific sites within the genome, you have to use chromatin immunoprecipitation. And you have to be very careful when dealing with protein and RNA samples, just because they degrade so fast. So making sure you freeze stuff quickly or get into some kind of stabilizing media rapidly is important if you want to have consistency and low noise in these approaches. And chromatin immunoprecipitation basically involves cross-linking your cells with something like formaldehyde. And what formaldehyde does is it sticks the DNA to the proteins that are next to it. So it just sticks things together. Then you take all of that, you basically blow it up. So you get a bunch of small fragments that you can pull down. Then you stick your antibody on a bead. Nowadays, usually that bead has a little magnet in it. And the pieces of histone that you're interested in, or transcription factor that you're interested in, will stick to these antibodies. You can use a magnet to pull all of those antibody-coated beads out of your solution, dump it off, clean it up, get rid of all the protein, and just look at the DNA associated with that particular protein. If you're just looking at one target, you can use a PCR-based approach. But if you want to look globally, you'll use a sequencing-based approach for this. This general sort of scheme of approach applies for a lot of different methods that are current in epigenetics. And again, there's so many different approaches to these questions now that I can't cover them all. But this schema of finding a particular set of things in the genome, pulling them down using an antibody to your target, and then sequencing the DNA associated with it is probably one of the more common methodologies, even if it's not specific to histones. It can be overall three-dimensional structure of the genome itself, et cetera, et cetera. And you can see here, this is sort of a global pattern of changes in some of the histone methyl marks in people with PTSD and without. And you can see that in a bunch of different cases, we have changes in the levels of H3K9 trimethyl here, this heterochromatic mark, changes in the level of H3K4, which is this high gene expression mark, K9 here, which is another heterochromatic mark, or K36, K9 here as well. So you can see this global change again in histone modification in PTSD patients. Generally speaking, levels of K9 trimethylation, which here correlate fairly well with levels of DNA methylation. The two marks interact with each other in the formation of heterochromatin. So this probably correlates with what I just showed you from Mehta et al. Paper from some years before, looking at the DNA methylation patterns, so that you do see this is essentially a validation independently that there is this difference in the methylation landscape in PTSD compared to control folks. Okay. Non-coding RNA is sort of the last major modification we go through. It's in some ways the easiest to analyze if you can get good samples. If you can get it frozen or treated in triazole quickly. You can do RNA sequencing or PCR, either one. Its actions are pretty complex, but finding it, looking at levels, quantitating it is pretty easy because PCR is such a common methodology. And if you want to look at it globally, RNA-seq is a lot more straightforward to do than, say, ChIP-seq, et cetera. But again, RNA is the least stable of all the molecules we're talking about. So you have to handle them typically by freezing immediately or by immersion in a stabilizing media like triazole. Then if you want to look at microRNA, which is probably the most common non-coding RNA to be looked at in clinical samples, then you need to make sure you're using extraction kits that don't filter out these small, less than 200 base pair RNAs, because a lot of the sort of standard off-the-shelf ones will do that. So microRNA actually degrade protein and mRNA and basically downregulate the targets. There are a whole bunch of tools to help you figure out which microRNAs might be involved in regulating the protein that you're interested in. Target scan is one of them. MIRDB is another one. Also, if you find a new RNA, microRNA, you can figure out what its targets might be using these tools as well. And this is a recent paper looking at microRNA expression. Apologize for the fuzziness of it, but basically what the group demonstrated is that people who were recently traumatized, there were several microRNAs that were highly correlated with the PTSD diagnosis. And you can see the stars for them here, even if you can't actually read that. Again, apologies. And that this class of microRNAs was involved in the regulation of FKBP5, which is this chaperone protein for glucocorticoid receptor that we mentioned previously. So this is just an example of how you can apply these microRNA tools in PTSD, how you can use computational analysis to do principal components analysis for a whole bunch of different microRNAs. In this case, what they did is they identified a bunch of microRNAs in the mouse model, and then they went and looked for those same microRNAs in blood from people who've been recently traumatized and achieved the diagnosis of PTSD. So that is one approach. Long non-encoding RNAs, generally we're looking at that in terms of GWAS or RNA-seq. This is an example here from a genome-wide association study. And you can see there's something right up here on the threshold that implicates this link RNA down here, AC 068718.1, which doesn't exactly roll at the tongue, specifically in female PTSD patients. And what you're looking at here is called a Manhattan plot. And this is typical sort of expression of hits in genome-wide association. And genome-wide association basically use uses very large populations of patients and controls to identify genetic loci that are associated with risk for a diagnosis. And so these lines that go across here are basically lines of significance thresholds. So this is sort of the minimum significance threshold here in blue, and this high significance threshold here in red. And you see that basically this one long non-coding RNA pops out here. So this is another approach you can take to discovering epigenetic actors in risk of disease. Is this GWAS-based approach. And more recently, there have been EWAS approaches where you look at the DNA methylome across these large samples of patients, hundreds of thousands of patients in some cases, to identify epigenetic modifications at specific loci that are associated with disease risk. Okay. So very few of these modifications have simple functional effects. I've kind of made it into a cartoon where it's up or down regulation of gene expression. And typically that is an output, but it is useful to look at those outputs and make sure that the modification is doing what you think it does. So doing some kind of measure of a functional outcome, whether that's a change in protein expression or RNA or cell phenotype or behavioral phenotype is desirable. If it's a local modification, usually just looking at the mRNA or the protein levels is the most useful thing to do. For global analyses, looking at RNA sequencing is usually the way that you do that. But of course, behavioral outputs, if you have a good idea that your epigenetic modification has a consequence for behavior, is another way to look at that. The big challenge with a lot of these more complex NGS-based analysis is the informatics. You're dealing with very large data sets and you have to use specialized tools to do it. You know, when I got into neuroscience, all I really had to know math-wise was a t-test and ANOVA. I've had to learn R and command line programming and stuff like that to start to analyze this stuff, or at least have friends who understand it. So doing these larger NGS studies requires some real bioinformatic expertise. Those tools are getting easier to acquire. They're easier to use than they used to be. But having somebody to kind of guide you through analysis is very, very helpful, because these are complicated and they're not skills that the average biologist, at least in my training cohort, had. Okay, so I hope I've made the case that epigenetic mechanisms are important to understanding of many of the lasting adaptations that are a consequence of stress. I've imparted some of the complexity of the subject, and hopefully given you a few thoughts about how to think about experimental design. I know this was a very top-level, broad overview of a very complicated field, but if you have any questions specifically about projects you're thinking about, we certainly have some time left and time for a break. If you want to stick around, or if anyone has any questions now, I'm happy to answer them.
I have a question.
Sure.
I've studied genetic epi a very little bit. They do GWAS and exome-wide and genome-wide studies, studies. Have you come across any literature that suggests that there's differences in ancestry based on how the DNA methylation changes?
How do you mean in terms of?
Like, well, I'm trying to think of one example we had in the class was that Asian populations don't metabolize alcohol. Some Asian populations don't metabolize alcohol in the same way other ancestries do. And I was wondering if stress had a similar reaction, if you've come across anything.
Yeah, so there's some interesting interactions with that way, and based on ancestries and genetic background. I mean, from the genetic perspective, like one of the interesting cases also involves East Asian or Southeast Asian populations where, you know, there's a specific mutation in the BDNF gene, which contributes to liability depression. So it's a conversion of a valine to a methionine at the 66 position on the BDNF protein. And that has an impact in European populations on the expression of BDNF. It's a lot lower, therefore there's less plasticity developmentally, and it's thought that lack of plasticity contributes to liability depression and drug addiction and things like that in later life. A lot of steps missing there, but that's the thinking. And then, but in some East Asian populations, there are mutations in an associated gene which basically compensates for that, so that you don't see any of the phenotypic differences that you do in BDNF expression in European populations. So yeah, those effects are definitely there. That's sort of one of the best described. You know, the alcohol dehydrogenase copy number variation is another one that you're alluding to in East Asian populations. So yeah, there definitely are differences in genetic background across human populations. It rarely segregates with something like race. It is more like an ancestry group, because again, I say East Asians, but it's actually much more specific to certain regions of Southeast Asia and certain ancestry groups within that where you see those particular effects. But yeah, it does seem to be the case that there is these kind of background effects. And in terms of epigenetic background, I don't know that anyone has actually sort of done EWAS or sort of global epigenetic landscape ancestry work as yet. I'm sure there are probably databases that you could query to do that. But one of the big problems, as with a lot of this stuff, is the weirdness, of the study populations. Like most of the people who volunteer for these studies, involved in these studies, are Europeans, in part just because places like the UK Biobank and Iceland have built up these big, and Sweden as well as a big twin registry, happen to be European countries. So like a lot of the big databases happen to be in highly European populations. So we really, as a field, need comparison from other human populations, because obviously white people are not the only people. I think we can all agree. So yeah, I hope that answers your question.
Great, thank you.
Examples that you gave were mostly about PTSD, which are quite, significant, drastic effects. Have there been any, epigenetic studies on more gentle kind of differences within the populations? For example, some of us study effects of stress during, pregnancy, or, just stress related to border migration, to autism. Any of that has been studied?
Yeah. I mean, I alluded to the little paper we had where we showed changes in mitochondrial epigenetics with people who had a history of childhood adversity and experience sort of lifetime perceived racism. There are other folks who've done experience of racism studies as an example of something that doesn't actually push people into clinical PTSD, but does seem to induce epigenetic changes. So yes, yeah, that is sort of an emerging literature. Obviously, as you say, PTSD is like a large effect size, and it also is a pretty pronounced pathology. So there's been a lot of interest focused there, just because it's easier to study, but also those people are perhaps more visibly distressed than some of these more subtle things that don't actually rise to the level of an overt pathology or a diagnosable pathology. But I think that, I think they all interact. I think that people who your sort of lifetime load of trauma definitely influences your liability to wind up with PTSD. So I think, yeah, all these more subtle things, people are starting to study them, but I'm really only seeing the papers really in the past couple of years. It's an area where a lot of work can and should be done.
Could you also comment about the sample size? Because, we submitted a couple of times a grant was 300 kids, 150, European Americans, 150 African American kids. And we already have RNA and DNA samples collected, and they had different exposure to metals and different cardiovascular and different psychological profiles. Nevertheless, it's been always shut down, not funded, because the size of the size effect. So anything that we need to keep in mind when designing our studies?
That's a big challenge, and some of that were you submitting to NIMH? Yeah, some of that has to do with sort of the policy outlook of the leadership in NIMH now, which I personally disagree with on a scientific level. I have no personal issues with them, but GWAS has become a major focus of what NIMH wants to do in terms of anything related to psychiatric genetics. And that means unless you have GWAS size sample sizes, they don't want to fund you lately, which I think is a very bad idea, specifically because of the kind of study you're talking about, which I think needs to be done and is kind of impossible to do at GWAS scale because those consortia don't exist for, say, environmental lead exposure. Yes, there's a PTSD consortia, there's a psychiatric genomics consortia, but I feel your pain is my point with that, but I don't know how to get past them. I think you might actually just sort of like go at NICHD, for child development, or try another agency, because yeah, it is an issue with NIMH in the past few years, is that they only really want to fund GWAS or GWAS validated research. So if you can find a way to write that grant where you can find GWAS like things that have been shown in GWAS, other GWAS studies as targets for your analyses, that might actually help it get over that hurdle. But yeah, this is my own experience, and it has been the experience of a lot of other folks who are sort of in these genomics adjacent areas of research, is that NIMH has a very specific idea of what it wants to fund as far as that stuff right now, and it is a challenge to kind of get around that policy prescription. So I am sorry. Any other questions? I think I'm probably past to let you guys go, but since we have a break, I'm happy to sit around and answer questions for a few more minutes if folks want to. All good? Okay. You guys should have my email. I'm happy to answer questions for many years to come if something occurs to you. And I hope this was helpful. And please let me and the organizers know if there's any feedback about it, things that you wish that I had covered, things you wished I hadn't, for future workshops. I really appreciate the feedback. And thank you for all your attention this morning. I really appreciate it. Ciao.
Okay, hi again, everyone. This morning I'm coming to you from my lab. I'm going to be working here this afternoon, so figured I might as well get started here in the morning. Anyway, we're on the home stretch now and going to be discussing the final topic, which is one that's relatively more recent to the stress field compared to the other biomarkers that I and other instructors have been talking about over the last few days. And so let me share my screen and then we will get going. Let's see. All righty. So we're going to be talking about the telomeres and how they can be used, or we believe they can be used as a marker of cellular aging and evidence that this can be affected by stress. So what are telomeres? So again, we're going to start at the beginning with some basic ideas, basic concepts to make sure that everybody has some fundamental information as we go forward into the use of changes in telomeres as a biomarker of cellular aging and again, affected by stress. So telomeres are, okay. Can you see my cursor? I'm not sure I'm on the right. No. All right, let me get over here. Okay. So can you see the cursor now? Not? Or not? Yes? Okay.
Yes.
Yeah, okay, thank you. So telomeres, so here's telomeres are on every chromosome. Here's a pair of chromosomes. Telomeres are at the end of the chromosome. They're capped and they're necessary for the maintenance of chromosome integrity, that is to keep chromosomes stable. So here is the telomere at the end of the chromosome. And there are proteins, family of proteins called shelterin proteins, which bind to the telomeres and help protect them from damage. And we'll talk about, as we go forward, we'll talk about some of the kinds of processes that can damage telomeres and cause problems, even with respect to vulnerability to disease. So here we have a cell that's replicating its chromosomes, replicating its DNA prior to dividing. So just like all the other DNA, all the other parts of chromosomes, which have to be replicated when a cell divides, the telomeric DNA also must be replicated. So here we look at number 2 here. The DNA polymerase is synthesizing the new telomeric DNA, but it can only replicate what we call the leading strand of the telomere over here. And the lagging strand, which is shown over here, has to be synthesized by a somewhat different mechanism that requires an RNA template. So unfortunately, this mechanism is not completely efficient, such that a certain portion of the end of the telomere is not included in the replication each time the cell divides. And as I say here, roughly 50 to 100 nucleotides are lost each time a cell divides, each time the telomeres have to be replicated. So human telomeres at birth contain about 10,000 nucleotides, and you can see this doesn't seem like much, but when you think about somatic cells that are dividing many, many times over your lifespan, not your neurons, but at least most neurons in the brain are not dividing, but your various somatic cells are. And so over time, the telomeres do get significantly shortened. So we're going to be concerned about this shortening. So we have to be able to measure telomere length. And there are 3 different kinds of samples are typically used in the studies of telomeres. So you could take a blood sample and isolate mononuclear cells, basically white blood cells. You can take swabs from the cheek, which are often used for other DNA purposes like forensics and identifying parentage, whatever it might be, or saliva, which does also contain actual cells, including epithelial cells and leukocytes, white blood cells, or tissue biopsy. Now, of course, for normal experimental research, you wouldn't be doing this. So you'd be likely using one or the other of these approaches. And here's just a bunch of measurement techniques, just to show you that there's different ways that different researchers who actually are engaged in this will use different methods for measuring. So this is a common one here. qPCR is quite common as ways that telomere length can be assessed. I think there's still a little bit of a holdover from Monday's long day, so I'm a little bit dry this morning. I'll try to keep my voice going. Now, here we're showing, cells dividing, and the idea is, again, just shown graphically here, that a little bit of the end of each telomere is lost with each division. So the telomeres are gradually getting shorter and shorter. Now, there is a process that can attenuate the loss of telomere length. Your cells contain an enzyme called telomerase, which can add on nucleotides to the end of the telomere. However, the telomerase is not fully efficient, not fully effective. And thus, even though this is to some extent counteracting telomere shortening, it still does not prevent telomere shortening over time. And this attrition, this loss of telomere length, can be accelerated by certain processes such as oxidative stress. If you're not familiar with that, we'll talk a little bit more about that in a little while. So because cells divide over and over again over time as we go on in our lives, we think about telomere shortening as a clock a kind of clock of biological aging. And this is the whole notion that if stress, bringing it into the stress and adversity field, if stress and adversity are perhaps causing us to age more rapidly in some fashion, this could be a way to assess this. So, and then finally, a final important point is if telomeres in a particular cell or group of cells become too short, this actually acts as a brake on cell division, and this is called cellular senescence. So this is the ultimate in cellular aging. Those cells can actually no longer divide. Well, this is just a cartoon to illustrate some of the things that are going on. Here's the telomere attrition that we've been talking about, the shortening. So as you might imagine, this, the rate at which this goes on in a particular individual can be affected by a number of factors. Some will be genetic, that you bring with you from birth. Others would be non-genetic, various environmental events, events in your life, particularly as we're focused on stressful or adverse effects, how you behave. Are you smoking? Are you engaging in other behaviors, drug use, that could contribute potentially to telomere attrition? And then as the telomeres get shorter and shorter, then there's evidence that this includes, leads to an increased risk for aging-related diseases and even perhaps shorter lifespan. So we can ask, is the shortening of telomeres simply correlative with disease, or is there evidence that telomere attrition could actually be causative, could actually be a causal factor in disease? And evidence for this, a causative role, comes from the discovery of telomere syndromes. So these syndromes are single gene mutations in the proteins that are necessary for telomere maintenance, including components of the telomerase enzyme. And these syndromes, they're rare, but they can occur in different organs and be associated with different disorders. And you can study this experimentally in mice by knocking out the homologous mouse gene. And as you can see here, the results do support a causative role. So telomere syndrome in skin cells, one outcome of this could be premature graying of the hair, bone marrow, leading to aplastic anemia, and so on and so forth, even including skin and blood cancers. So now we get to the nub of the issue for us. So I've already shown you how telomere attrition is something that occurs during aging, but are there other factors just besides aging itself, just besides time itself, that can influence telomere attrition? And a number of studies over the last, say, 10 years or so have begun to really examine this. So the majority of these studies are cross-sectional, where you look at individuals, say at different ages, but also importantly, individuals who have undergone different degrees of stress or adversity in their life. And you look at differences in telomere length. And if telomere, for example, if telomeres are shorter in the individuals who have been stressed, then that suggests that the stress contributed to a shortening of telomeres. Even more powerful, I think, are studies that are longitudinal, that actually assess, that sample telomeres in the same individuals and look for actual evidence for shortening, whether shortening is occurring faster in some individuals than others in relation to stress, adversity, et cetera. And both human and animal studies have been conducted. Of course, we're mostly going to be talking about human studies, but a little bit of animal stuff where it's relevant. So right off the bat, I liked this meta-analysis. It is based on animal work, and it's a meta-analysis of a number of studies looking at various species of non-human vertebrates living in the wilds, So these are not animals kept in the laboratory, but rather animals that are caught, and their telomere length is assessed, and the researchers have examined various kinds of environmental stressors that could be affecting animals' health and so forth. So you see examples here, harsh abiotic conditions, activity level competition among animals in a species, human disturbance of habitat, pathogen infections, a whole bunch of different things. And so these are show the degree of estimated telomere shortening with the 95% confidence intervals. And this is the overall effect size. So there's a lot of animal studies looking at natural stressors that are supportive of the idea that chronic stress is associated with more rapid telomere shortening. All right, I apologize for this. I don't like to apologize for slides, but this one warrants it. This is a meta-analysis from a few years ago looking at a variety of different kinds of human stress exposure. And in this case, the various studies used a correlative approach, correlating the degree of exposure to these different kinds of stressors with telomere length. And don't worry about all the numbers here. You can just look at the graphic. So this is a physical disease. You can see a significant effect here. This would be the 0 here. Again, confidence interval. So as long as the confidence interval does not overlap 0, it means we have a significant effect. Psychiatric illness in this meta-analysis was associated with telomere shortening. Smoking, alcohol use, there's a slight trend, but it wasn't significant. Psychosocial stressors, decent effect size, and so forth. So there's no overall, in this case, there was no overall effect size estimated, but you can see that a number of different kinds of stressors were associated with significantly shorter telomeres. Here's another meta-analysis from different authors. And in this case, they looked at perceived stress. So of course, this is interesting because the previous one, we're actually looking at assessment of the degree of stress. We didn't have a different biological measure, just the telomere length. But here with perceived stress, we see that most of studies did not find a significant association between telomere length and perceived stress, except for one study over here that had quite a substantial effect. There's a slightly significant, just very small effect size here overall. So there was still this mathematically, there was a small association of reduced telomere length that's going here to the left with perceived stress, but it's really not robust at all. All right, so now we're going to look at a few individual studies as well. And the first one is a pretty recent study using the strain lifetime assessment of stress and adversity. And participants were 183 women. And interestingly, the authors chose to assess women who were mothers of both neurotypical children, but also a number of them were mothers of autism spectrum disorder children. And I think that the reason for this was to try to include in the group, in the assessment, individuals who would be facing the additional challenge of dealing with and raising kids who were on the spectrum. So the strain was used to assess lifetime exposure to stress and adversity. And this was a longitudinal study, which I said, I think are powerful. The women were, telomere length was determined 2 years apart. And so you could get an indicator of baseline telomere length and whether that related to scores on the strain, but also the attrition rate. How much were telomeres shortened between the first measurement and the second measurement 2 years later. And so the first result was that on the strain, a higher score for chronic difficulties, just chronic life difficulties, predicted lower baseline telomere length and more rapid shortening when they looked at the two measures. And this is the attrition. So this is on the y-axis is the amount of difference between the first and the second measurement. And then this is the total count of chronic difficulties across the lifespan. And so you can see the relationship there, which was significant. From the same study, they decided to look at the data in a couple of different ways to parse the data a bit. And one of the things that they chose to assess was, again, for these women who were, in this both neurotypical and autism spectrum disorder kids, but they asked in the strain, you could indicate where in your lifespan you were encountering these various stressors or various life difficulties. And it was quite interesting that the rate of telomere attrition was actually affected the most by the chronic difficulties that the women had experienced in their childhood, especially their early childhood, even more so than adulthood. So it doesn't say that there was no effect of difficulties that they were encountering in adulthood, but that there were lingering effects of early childhood difficulties that were now showing up in rate of telomere loss, length, loss of length in these adult women. So we're going to come back to this idea of early adversity, and we're going to see more and more of that as we go forward. So here we have, whoops, oh, this page cannot be displayed. Ooh, that's unfortunate. I don't know what went wrong here. It was fine on my computer when I looked at it earlier this morning. Well, anyway, so I'll just describe it without the graphic. So you can see that over 4,500 adult participants were assessed in a large study. It's a US health and retirement study here. And they were assessed for childhood and adult adversities and whether those are related to telomere length. And there were adjusted odds ratios. So this was yes or no. Do you have shorter telomeres or not in relation to any specific adversity? And I don't remember, I don't have the graphic to help my memory. I don't remember what the childhood one was. The specific adult adversity was rather interesting. That happened to be need for use of Medicaid. So participants in the survey, again, it's a health and retirement. So you're looking at partially a older cohort of individuals. And if particular participants in the survey, if they had needed to make use of Medicaid, then they had an increased risk of having shorter telomeres. And so what does that mean? You know, when I think about this, I think that needing Medicaid is a proxy for two things together. I think it indicates lower SES, because Medicaid is directed towards helping elderly people, retired people who need healthcare, but also the fact that you need it indicates perhaps that you have poorer health. So I think those two conjoined probably are what leads this outcome. So there are others, other factors were in the predicted direction, but only these two, including this one adult one, turn out to be significant in terms of the odds ratio. So then, again, looking at early life adversity, a meta-analysis that was done in 2018 or published in 2018. So each one of these, I'm not sure I made it clear before when we looked at the depiction of these meta-analyses. For those of you who might be less familiar with it, each one of these squares with error bars represents the outcome of one study, okay, one particular study. And then the meta-analysis statistically combines them. They're weighted with respect to the number of subjects, number of participants in each study. Again, just to go back and give you a little bit of grounding in meta-analysis. So anyway, so the ages encompassed in the studies that were assessed here in this meta-analysis went from prenatal stress to 18 years of age. And one of the things the authors did was to group effect sizes according to the period of adversity. So early development, including prenatal up to 4 years of age, what they call childhood, 5 to 12, and then adolescence. And the greater effect sizes were yielded by having stress and adversity during the earlier periods. However, if you disregarded the age here, the age range, but rather analyzed the data for the time between the experience of adversity and the assessment by the researchers, more recent adversity yielded greater effect size. So that may, in some sense, seems a little paradoxical or contradictory, but I think we can conclude two things. Number one, that early adversity can cause or be associated with, be careful here, but be associated with lingering effects on telomere attrition, telomere shortening, but also that in addition to that, that recent exposures can additionally speed up the process. All right, so I'm going to take a moment here to allow you to raise questions about any of the things we've talked about so far. Any questions?
I have a question just based on those last few slides.
Yeah.
So do you think it'd be really valuable to collect measures of chronic stress from like historic, having adults recount their chronic stress in childhood as well as their kind of more concurrent chronic stress when looking at these things in adulthood, or how would
Yes, I do agree. I do think that is useful. Some of these measures, of course, do attempt to get at that. We always need, of course, as you know, we always need to be aware of recall bias and issues with retrospective self-reports of stress or other, life events. But absolutely, I do think that it could be a very valuable addition to the assessment of recent or current stressors.
Jerry, in regard of allostatic load, I've never seen any studies that incorporate telomeres. Are there any?
Yeah, are there any? I'm trying to recall. I think there might be, but I can't say for sure. I can't think of a specific reference to, to give you off the top of my head. I think I have seen that, but it's been rare.
Yeah,
I think we might expect to see, since the field is growing and more people, that telomere length still, telomere measurements are still the province of a relatively small number of labs. At least the papers that I see, the impactful papers, are from a relatively small number of labs. The labs that are really doing good work and are sort of making the headlines, if you will, in terms of strong results. But I think that we are likely to see more incorporation of that as a biomarker, and it may turn out to be a very good one. Remains to be seen.
thank you.
If you could just give me 1 second, I've got to run to the lab's water jug and just get a quick drink. I'll be right back. Okay. All right, then let's continue on.
Could I ask a quick question?
Yeah, sure.
Is there evidence for things that can slow the attrition or?
That is a great question. I'm going to, yeah, but I'm ahead of you because I'm actually going to take that up briefly at the very end.
Great, I assume. Thanks.
But it's a great question. I promise everybody this was not a planted question. But yes, I am going to address that a little bit because, we have to think about that in our own lives. If we know that this clock is ticking, and when you get to be my age, it's really ticking. So we'll, as I say, I'll talk about it a little bit, and there's not a lot of research on it, but I'll bring it up. And if you want to ask more about it, then feel free. Okay, so let's continue on with this theme of chronic stress or adversity relatively early in life. So another review, in this case by Coimbra and others, 11 studies that examined exposure to adversity and as well as measuring telomere length in children. See the age range and the different kinds of approaches. And what was interesting in this review was that shorter telomeres were associated with lower SES, living in a disadvantaged neighborhood, duration of institutionalization. So I suspect that many of you have heard of or know about the famous, it's still ongoing actually, studies of Romanian children. This was during the infamous Coescu regime in Romania, when large numbers of children were kept in terribly impoverished conditions in state orphanages. And researchers have had access to these children, are now adults. Quite a bit of time has elapsed since then. But some of these individuals, these impoverished, raised kids are still being studied. So by various research groups. Anyway, duration. So the amount of time that individuals were kept in these terrible orphanages, exposure to interpersonal violence, and also exposure to maternal clinical depression. So this is just to show you a range of different kinds of adversities experienced during childhood that were found to impact telomere length. Now, what about the flip side? What about resilience? This is a very recent study that I noticed. So this study just came out earlier this year. It was done in Finland. So a nice, good size sample size. And the authors took multiple measures of maternal mood and also assessment of stress during pregnancy. And then they calculated a resilience score. If you're interested exactly how they did this, I recommend the paper. It's in my folder. So they looked at stress and resilience, and then they measured telomere length from white blood cells, leukocytes, obtained from cord blood, okay, at the time of delivery. So cells from the fetus are sloughed off and get into the umbilical cord. So you can sample this and get an indicator of telomere length of the newborn baby. And so there were two main findings. I'm only showing you one graph, so I'm not showing you this graph, but maternal stress was inversely related to newborn telomere length. So the greater amount of maternal stress tended to be related to a lower telomere length in their babies, newborn babies. But on the positive side, since they also measured positive indicators in the mothers, higher resilience score, these are quartiles, higher resilience scores were related to a longer length of telomeres. So, so stress, this is one outcome. There are many others, but this is sort of the newest outcome of how maternal stress gets transmitted to the fetus and actually affecting telomere length in the fetus. Now, of course, this is the first study that I know of this kind, so obviously we need replication and so forth, but it's quite intriguing. So Alyssa Eppel is one of the outstanding researchers in the telomere field. I highly recommend her work. And we're going to see a few papers of hers cited here. But this is just a cartoon from this review of intergenerational transmission of short telomeres. So we already saw a piece of evidence for that in terms of maternal stress and resilience affecting telomere length at birth. But in addition to that, stress and adversity during the lifespan affect the rate of telomere attrition. So one of the points that Eppel and Prather are making here is that what wasn't assessed in the previous study, but what has been looked at is the amount of adversity that mothers were exposed to during their childhood, probably their mother's stress as well, but during their childhood and adolescence, have become more vulnerable to stress and mental illness during their pregnancy, and then these effects are passed on to the offspring. And this is just a little cartoon. Perceived stress will, we've already seen there's some relationship between perceived stress, measured depending on how you measure it, with physiological arousal, HPA, axis activity. We're gonna actually talk a little bit about the HPA axis in relation to telomere length as one mediating factor, perhaps. So, and then affecting telomere maintenance and how telomeres changes in their length, how rapidly they shorten over time. So this is a slide that's especially attuned for the biologists and biochemists in the group, but I'll explain the key points here. So this was, the author here was actually focusing on animal research, and that's why the main glucocorticoid being shown here is corticosterone, which is the main one in rodents and birds, but also mentions cortisol here. So these are enzymes that are among those that regulate what we call oxidative stress in cells. So oxidative stress is produced by something that's occurring in all of our cells to varying degrees depending on the metabolic pathways in a particular cell. Some of the reactions that give rise to oxidative stress produce hydrogen peroxide. So that's what this is here, hydrogen peroxide. And hydrogen peroxide can be converted to hydroxyl radicals that's shown here. So this is oxygen, this is hydrogen, and the little dot here is a free electron. So these hydroxyl radicals and other radicals, this just means, another part of the molecule. There may be peroxides here with 2 oxygens with a free electron. These guys are bad. They're bad in your cells. They're highly reactive. They can damage various cellular constituents. They can damage nucleic acids like DNA and RNA. They can damage cellular proteins, and they can damage cell membranes by affecting lipids. So they call it lipid peroxidation. So as you can see, again, all of those things are inimical to healthy cells. So you have various mechanisms within cells, since these are being generated, to get rid of these guys, to inactivate these guys. But to the extent that they're produced, again, they can produce cellular damage, including, remember I mentioned nucleic acids, I mentioned DNA. So these guys are thought to be able to cause telomere damage, including increase in the rate of telomere attrition. So it turns out that certain biochemical pathways that are regulated by glucocorticoids can increase the rate of free radical, oxygen radical formation, thereby increasing oxidative stress, and thereby causing an increase in the rate of telomere attrition. So a second potential mechanism, as shown by this arrow here, is effects of glucocorticoids on telomerase activity. So the only thing that I could find on glucocorticoids directly affecting telomerase was an in vitro study, so a cell culture study done a while back. And the authors exposed mononuclear cells and white blood cells, so typical kinds of cells that would be used to measure telomere length, to various concentrations of hydrocortisone, which is the same, just another word for cortisol, for 3 days. So the cells are constantly being exposed. This is not a physiological condition, because obviously the amount of cortisol in your blood is changing with rhythm, it's changing with stressors and so on. So this is a constant, pretty constant level for 3 days. And then they actually measured enzymatically telomerase activity. So low cortisol levels, not shown here, had no effect. Higher levels, you can see this is a log scale here, showed a gradually reduced activity of telomerase. Now, I really want to emphasize that levels above about 1 micromolar here are not physiological. And that's important. And I can tell you that the other area of my research, which of course I'm not talking about in this workshop, is pharmacology and neuropharmacology. And I can tell you that people who do these kinds of in vitro studies, cell culture studies with drugs, are hammering cells with huge concentrations of drugs and don't seem to pay attention to whether the results are translatable to a living organism. So I always, recognize that you need to look at concentrations carefully when you're looking at any in vitro studies to make sure that what they're doing at least has some relevance for the accurate physiological condition. So here, yes. So I think we can see that within the physiological range, there is some modest reduction in telomerase activity, which would tend to perhaps enhance telomere attrition, since the telomerase is counteracting that and trying to keep the telomeres longer. So mostly we've been talking about chronic stress, and we can ask, is there any evidence that the magnitude of acute stress responses is predictive? So I'm not here saying that a single acute stress response is going to affect your telomeres, but rather is the sensitivity of your stress response system. Could this predict the rate of cellular aging that's occurring as measured by telomerase attrition? So I thought this was a rather intriguing study. They were looking at early aged individuals and using salivary cortisol responses as their index of acute stress reactivity. And they had two different kinds of stressors. So for those of you who have a psychology background, you will know about this, the Stroop color word interference task. So you see the subject is given a bunch of words in a color that's discrepant from the name of the word. So when you see this word here, orange, your first inclination is to say orange because it's the word orange, but the correct response in the test is to speak the name of the color. So these would flash up on a screen, let's say, one after the other. They're not actually shown all a bunch at once. And your response time, how fast you can correctly give the color and not the word, and also how many errors you make, would be an indication of your accuracy on the task. But here it's meant to be a stressor because you're trying to do it correctly. And the second stressor is a mirror tracing task. So this is another mildly stressful task. You have down here that you can't directly see is typically a star-shaped pattern, like a 5-pointed star, and made up of 2 parallel lines forming the star. And you have to trace accurately within the 2 lines, not touching either of the edges, using a mirror, which inverts the image and is tricky to do it correctly. Anyway, so those are the tests. And so when they looked at here's the pattern of salivary cortisol. So there were a bunch of responders here. So these are the responders here, the increase post-task and then recovery. But then over 200 were non-responders. So they weren't ruffled by the task at all. There was no increase in cortisol. So then, since this is a longitudinal study looking at is there any predictive power of your stress responsiveness to telomere shortening? They looked at this longitudinally and they got telomere measurements again 3 years later to be able to compare the 2 groups. So here are the results. So here are the stress non-responders are here. This is the amount of shortening over 3 years. And here's the stress responders. Very little difference at baseline. And here they are, significantly greater rate of attrition over 3 years in the individuals who had responded to those stressful tasks. So this supports the hypothesis that greater stress responsiveness is associated with a more rapid rate of telomere attrition. But the outstanding question, the question that's not answered here, which the authors acknowledge, is you would think that if this stress responsiveness or non-responsiveness had been going on previous to the beginning of the experiment, why didn't these guys show a difference at baseline? because they obviously didn't. So, and the authors don't know, and we don't know. But at least with respect to this 3-year period, the difference in responsiveness was predictive of telomere attrition. So going now back to the HPA axis, we can ask whether there's a relationship between HPA axis activity measured in other ways, apart from stress responsiveness, that we looked at a moment ago. And a little bit older study looked at this in adult women. This was a small N study, so really kind of a pilot study. And the authors looked at nocturnal urinary cortisol excretion, salivary cortisol responses to acute stress, and also slope, which you remember we talked about on Monday morning. And if you compare the lowest quartile versus the highest quartile, you see that the telomere length was shorter in every indicator of HPA axis function in the individuals who were high in that parameter compared to the lowest quartile. Some of you are probably interested in whether telomere length is related to psychopathology. So this was reviewed a few years ago by Lindqvist et al. And I have no graphics here to show you, but rather just some conclusions from the study, from the review, that there were relatively consistent associations found for several different disorders or categories of disorders, including major depression, anxiety disorders, and PTSD. And this is, we don't have to go into this, but this is just a cartoon showing various pathways by which psychopathology, so perhaps mediators whereby psychopathology can lead eventually, not showing the biology, but rather this is more the psychological aspects associated with psychopathological disorders, how that may converge to lead to telomere attrition, and then this in turn being associated with increased risk of various chronic diseases. So now we're back to a couple of meta-analyses looking specifically at depression, since review had indicated that there was a relationship between depression and telomere length. So this is a little bit interesting because both of these reviews and meta-analyses were published in the same year. They did have 14 sources in common. The Lin, which had fewer, the Lin is over here, had fewer sources, but did have 2 that were not in the Ridout review. But Ridout included 24 that weren't included in Lin. So their criteria for inclusion differed enough such that there were many more studies included here. The effect size was greater here's the overall effect size here. Again, individual studies, shorter telomeres here, longer telomeres here, same thing here. Here's our overall effect sizes, greater with the smaller number of studies. So, at least they did come to the same conclusion, but it looks like that Ridout was including a bunch of studies that had really showed little or no effect at all of a relationship between depression and telomere length. So the Eppel and Prather review looked at some of so what was nice about this was they reviewed both human and animal studies. We saw a little bit of that earlier. on relationships between telomere stress and depression or depressive behavior. And so some of the factors that are important in this relationship include genetic differences in vulnerability to stress, starting over here. Of course, high levels of stress exposure. And then telomere biology, low telomerase would contribute to the ability, that is telomeres not being able to be recovered, their length recovered as readily, greater rate of telomere attrition. So again, if you want to look at this when you have access to the entire thing to look at the details here, you're welcome to do that. All right, so most authors of research on psychopathology, on psychosocial stress and so forth don't address the question of whether the results that we see using peripheral cells, which is necessarily in human studies, necessarily what we have to do, whether those measurements in peripheral cells are reflective of what's going on in the brain. So this right now is basically an open question. So keep in mind that here, I think one reason why perhaps this has not been a main topic of discussion is because of the fact that there's only relatively just a few populations of dividing cells in the brain. So as I said, that is dividing neurons, sorry. So, but there are, and they're important. So there are dividing cells in the brain, germinal zones that give rise to new cells that are incorporated into the hippocampus, very important brain area for many functions, including memory and learning, and others as well. And also there are dividing cells that contribute to a neuronal turnover in the olfactory system is another example, but also glial cells in the brain divide. And glial cells do affect, have relationship with the neurons in ways that can affect our behavior. So the point is that cell division in the brain and possible, impacts on telomere shortening is a legitimate topic, but has been understudied. And we would have to do this in using controlled animal studies. And I think it's something that is worth people investigating in the future to see whether, and then of course, relating this to processes that affect telomere attrition in peripheral cells, to see whether the two are tending to be related to each other so that the peripheral cells are predictive of what's going on in dividing cell populations in the brain. All right, so now the very last here is, can stress-related telomere attrition be prevented or reversed? So a question was brought up earlier. This has been studied a little bit, but not really very extensively at all, and not very well controlled studies. My reading of what literature is out there is that there is some evidence that telomere attrition can be counteracted by healthy behaviors and perhaps by certain kinds of interventions, such as mindfulness-based interventions. But as I say, the research is sort of right now what I would consider pilot stage. So we'd like to see a lot more work done of that kind to tell us, are there best practices? Are there things that we can do in our own lives that would, not only help us in other ways, all these things are good for our health in other ways, but whether this also contributes to a reduction in the rate of cellular aging as measured by telomeres. So this is just fine. Again, there's a lot of stuff on this slide, and it's a summary slide. So again, if you're interested in this and want to go through this, all the details of this later on when the video is posted, then you're welcome to do that. But here's our conclusions. So what I've shown you is that telomere length is used by biologists in many fields, certainly the field of aging itself or senescence. The telomere length can be thought of as a biomarker of cellular aging, but there's more that goes into it in terms of the fact that there's now strong evidence, as I've showed you, that telomere attrition, the rate of telomere reduction in length, is exacerbated both by adult stress, chronic stress and adversity in adulthood, but even perhaps more so by adversity early in life. We've also seen associations between shorter telomeres in individuals suffering from various kinds of psychopathological disorders. And we hope that research will enable us to know whether healthy behaviors can slow down or even perhaps reverse telomere attrition. Probably not the latter, but at least slow it down. And we've seen there basically in the context of the workshop is that telomere length, I think, can be a useful biomarker in studies of chronic stress and adversity. So that's it. Open for questions. Everybody all wiped out from 2.5 days of intensive work.
So just a quick question then.
Yeah, could you speak up? My speakers here in the lab are kind of low volume, so please speak up. Yes.
Is it better?
Yeah, it's better.
Okay. So kind of tying the two talks together, has there been a relationship or it just seems like epigenetics and the telomere length kind of go hand in hand. Have there been any studies kind of looking at these two and kind of like what the interplay is? And and this would lead to the question then of epigenetic changes can then have effects on the telomeres, whether it's be in their maintenance or their lengthening.
Yeah, Richard, could you address that?
Yeah, I mean, it is a very interesting and I think understudied interaction. You know, a telomere itself is just not it's not just these repetitive sequences of DNA. It's also a heterochromatin cap. So part of how it survives under good circumstances is it's got this kind of protective nucleosomal coating like the H3K9 trimethylation and DNA methylation that sort of keeps it stable and protected. So the same mechanisms that are involved in silencing other parts of the genome that are more active are present at the telomere. And in fact, some of what we know about how heterochromatin is maintained during cell fate comes from our understanding of what RNAs called teras, which are non-coding RNAs that telomeres produce, do to like nucleate heterochromatin regions, because it turns out there are other non-coding RNAs that do the same kind of thing in terms of cell fate and differentiation. So very similar mechanisms are involved. And I suspect that, you know, things like nutritional deficiencies in methyl donors or things like that, where you're competing with other parts of the genome for the things that you need to keep us, you know, a properly heterochromatinized telomere cap, probably contribute to some of this degradation of telomere length and things like that as well. Similarly, I think that there's probably interaction between some of the genes that drive epigenetic change, like the steroid receptors and telomerase. So I wanted to ask Jerry this actually. Has anyone actually looked to see if there are glucocorticoid response elements anywhere near telomerase in the genome?
Yeah, it's an interesting question. I should follow up on that and see, because offhand I don't know.
Yeah, I haven't seen it either, and it's challenging. One of the reasons that's hard is you can't just look in the promoter right close to the gene, is that glucocorticoids, as it turned out, tend to have their affects very distally as part of enhancers more often than not. And so you got to look a good distance away to find it, which might be why no one's done the easy study yet and just, you know, seeing if GR sticks somewhere close.
Yeah. Other questions? So the schedule calls for a break, and you know, this the schedule is kind of was kind of copied and pasted from a few years ago when we were doing this in person, and we would actually take a break and then come back as a group in person to have an end of the workshop Q&A. Doesn't make any sense to take a break now. So I think I will just ask if there are any overall questions about workshop, any topics. I mean, Richard and I are not really in a position to answer any questions about what you guys heard yesterday. But if you want to, if anything occurred to you relevant to either of this morning's presentations or Monday's presentations, feel free.
I have a quick question, please. When you were discussing how you could use the nail and the infant to get at the prenatal environment, the stress?
Yes.
You had mentioned about, you know, the nails, they're not going to really have them right away. When is a good time to try and get them? Like a month after they're born, or?
Right. That has not been systematically examined. Prior to the use of nail sampling to assess steroid hormone accumulation prenatally, nails were already being used a little bit for exposure of infants to substances that, you know, toxicants that the mother had ingested during pregnancy. And unfortunately, in those studies, the nail collection of the infants was not done systematically. To my recollection, the authors would the researchers just ask the parents to collect nails whenever they were going to collect the nails, you know, keep the nails when they happen to feel like it was appropriate to cut the nails. So this was not done systematically, and I don't think it's really been done systematically even yet in the hormone field. I will say that we think, based on rates of nail growth, both prenatally and then postnatal in the neonate, that probably a couple of nail collections. Again, I can't say exactly. Again, some kids, you know, some babies will grow their nails a little bit faster than others, obviously, but probably a couple of nail samples taken postnatally would still mostly reflect prenatal accumulation. So, but this is based partly on supposition and not with empirical proof of it.
Right.
So it's something that, it's something that definitely needs to be studied more carefully and to be systematized in a way that we're starting to try to systematize nail collection in adults.
Right. So you had said that they grow the nails at different rates. So maybe like a couple weeks out, or
probably, you know, probably like a month out, and then maybe another month later. So, you know, again, you think of the nail being formed. I'm trying to remember the term for it, where the nail originates. I don't remember, but anyway, the time it takes for the nail to begin being generated and then to work its way all the way down the finger This is going very slowly prenatally, and of course the finger is very tiny in a newborn baby. But we have done newborn nail samples. When I say newborn, I don't mean, of course, at birth, because there's nothing there you could really get, but without harming the baby. So, but families that have collected nails just in a non-systematic way, just whenever it seemed like the nails were long enough that they wanted to cut the nails. And we've had families collect nails in just a small pilot study, unpublished, for, let's say, 2 times. And it gives ample, the nail levels like newborn hair is quite high. So it gives us enough material to measure the cortisol concentrations in the nails. So, but how much of it is prenatal and how much of it is postnatal is not entirely known. So you're getting, I think you're getting an index of that's at least partially prenatal, but maybe confounded by postnatal events as well, postnatal cortisol exposure as well. Just to add one more thing to that, the reason, I think I mentioned a little bit on Monday about the reason why we're suggesting, possibly using that approach is because I gave some of the reasons why studying stress and cortisol levels in the mom has, severe limitations, I think. Yes, it's telling you a lot about the state of the mother's HPA axis, but there's too many other things that are intervening or confounding that in terms of how much cortisol the fetus is exposed to. So at least by taking an offspring measure, you are keeping in mind the limitations. At least you are directly getting some kind of measure of exposure of the offspring themselves, and not just what's been going on in the mother, independent of the baby's own or the fetus's only cortisol release, effects of the placenta on cortisol metabolism, and so forth.
I had one quick question
Yeah.
about nails and getting samples from adults and just getting the regular nail clippings.
Yep.
I'm just wondering if the length of their nail beds makes any difference in terms of the clippings you're getting. And
yeah, it does, and that's that's a reason I mentioned that there are differences between the different fingers, and the rate of nail extension is a little bit greater in longer fingers, and the nail bed is also longer, as you might expect, in longer fingers. So but the average, if you look at the average across all the fingers, it's about 1 millimeter per 10 days.
Okay.
So we, at one point, we were trying to standardize getting 2 millimeters of length. And if you want to do it that way, there's actually a really easy way to do that. It turns out, which I happened to get from a drug company that was doing forensic analyses on nails for drugs. And it turns out that the thickness of a standard US quarter is about 2 millimeters. So all you need, you don't need a ruler, a metric ruler. All you need is a quarter. Take it out of your pocket, and that gives you about, pretty close to 2 millimeters. So if that's what you wanted to do, you could use that 2 millimeters. But I think it's better to do the clipping and then reclipping and wait a specified period of time between the two. And so then you can specify. But either way, you can specify something. You can either say we've got 2 millimeters worth of nails. We know that nails grow 1 millimeter over 10 days. And so it comes out to about the same thing, if you think about it. If you have 18 to 20 days between clippings, then that should give you about 2 millimeters of nail clipping, right? So 2 different ways of getting, skinning the cat, if you will.
Okay, thank you.
Yeah.
Hi,
I don't know. Sorry.
Oh, thank you. I had a question about hair cortisol. So when we're talking about hair cortisol, you mentioned how I think it was the perceived stress scale like didn't map on to the hair cortisol, what the hair cortisol was finding. And I was just wondering if you could kind of speak to that. Like how do we reconcile when the subjective self-report measures don't necessarily map onto what we're finding with these biological measurements?
Yeah. Well, first I should clarify that the PSS was not predictive or did not significantly associate with hair cortisol in the Weckwesser 2019 paper, in contrast to the Hassel's scale that they used in that study. If you look at the rest of the literature, as many people have used PSS, and some studies did find a significant relationship, statistical relationship between the 2 measures in their group of participants, but others did not. And so overall, the literature has been inconsistent. And so for sure, we cannot say that measures of subjective stress are consistently related in magnitude to measures of adrenal output as indicated by hair cortisol. For sure, there's no consistent relationship. And I think that you can think about that in 2 different ways. One is that each person brings into the situation, the context in which they're being stressed, they bring into it a variety of factors, some biological, their genetics, their epigenetics, as you learned a lot about this morning, their previous experiences, their upbringing, their previous experiences, their psychological traits that they have. And though all of those factors that influence your stress response, that influence, plus external things like, your social network for coping. So we need to think about those things too. All those things, we believe, based on the data that we have, can differentially affect how strongly you respond to a given stressor psychologically, subjectively, versus how much you respond physiologically, whether it's measured by cortisol or whether it's measured by any other kind of marker. Cortisol's, used most frequently for looking at physiological responses to acute, in addition to things like heart rate and blood pressure, which are also widely used. So all of these things can differentially affect these 2 manifestations of your response. Your subjective manifestation, what you fill out in a questionnaire, whether it's PSS, Hassel scale, strain, whatever, versus your physiological response. And that's why we have this workshop. If all you needed was to give your subjects a few questionnaires, that's a lot easier and cheaper, right? I mean, you sit them down, you give them some questionnaires. This is how stressed you feel. This is, what's been going on, and this is how depressed you feel. This is how anxious, and so on. And and of course, people who are just psychologists, that's what they do, and that's great. But the point of this workshop is we have to look at the biology too.
I wanted to make a comment, if I may.
Sure.
I really like the presentations of a variety of researchers and that we had during this workshop. In particular, when the data is presented, also critically analyzed and weaknesses are shown. That was very, very helpful to me in making decisions. On the other hand, even though you asked us not to talk about yesterday because you weren't here, but in the afternoon presentation when we had a private company presenting their instrument and plates, I perceived that as very biased. So I would like to consider maybe finding a researcher for that section that could present a variety of options for us to consider rather than single company talking about their wonderful product. Something to think about. I mean, I'm not saying that it shouldn't be done, but maybe it would be a little less, less one-sided. Let's put it this way.
So, so let me address that, and I think that's a valid point. I think the value of having a company come in is because the researcher who's giving that presentation, who is an expert in the field, has found that this company's product, that their equipment works well and gives them high quality data. So that, and this is therefore perhaps a starting point, a recommended starting point for researchers who might want to incorporate that kind of technology, that kind of assessment into their research. On the other hand, I think you raise a valid point that there's always, competing companies and alternatives. So I thank you for raising the issue. And what I can suggest is that Katie and Lisa mention, some of what they consider to be good, reliable alternate sources, other companies. So, I talked about in the cortisol, I talked about several different assay methods and ones that I thought were good and others that, like the Alpco kit in particular for hair cortisol. But they could do the same, at least a little bit, so that perhaps it won't feel biased in the way that you described. But we did have a reason for doing it the way we did it. For people new to the field, I think it's good to have advice from an expert. You're free to use it or not, as in anything, but it's a good point and one that I'll bring up with them.
Can I ask one more question?
Sure.
So in thinking about the differences between more perceived and physiological stress, what are kind of your ideas around how those differentially predict behavior then? Or do you think it's right to then think about, okay, perceived stress tends to be predictive of these sorts of things and physiological predictive of these sorts of things, or how do you?
Yeah, that's a very complicated question. And one that I'm not going to try to answer. I think that, for any individual study, if you have a complex design and many outcome measures, many variables that are affecting your outcome measures, and then a number of outcome measures, I think the sensible thing to do is to put all of those things. Now, of course, you need to have a good sample size if you've got a lot of variables, to have adequate power to test your hypotheses. But I think that what makes sense is to plug the variables that you're taking, demographics, potential confounders, all kinds of things that you have measured that your participants, your subjects are bringing into the situation and into the study. And then you've got a variety of outcome measures, some psychological, subjective, some biological. You put them together into your complex analysis, and like a multivariate analysis, and you determine empirically what are the predictors of perceived stress, what are the predictors of cortisol responses, are they the same, do they interact, do your quote independent variables, if you want to call them that, how do they interplay to affect the two? And then you may find that there's overlap. You may find that there are differences in how these quote independent variables are affecting the two different kinds of outcome measures. But there's no like one answer that, and again, that's why I never say that one is better than the other. It's not that one is better than the other, or one is more predictive than the other, or can be predicted more than the other. They're giving you different information, and can well be, as we've seen, differentially affected by the various factors in a study.
Yeah, just to comment on that. I mean, I think that is kind of the question for a lot of this work is how do you relate these extrinsic factors to the physiological outputs and to the behaviors? And, you can imagine any number of things, whether it's culture or, big five personality traits, and people have sort of worked on all those different aspects of it. I think, from my perspective as someone who's really curious about the discovery, I think what Jerry is saying is design good studies to get at these kind of things. Like what do externalizing personalities tend to show in terms of the correlation between self-report and cortisol levels, for instance, or what do cultures with low emotional expression show relative to with high expression, because just to name a couple random variables, that have come up in my thinking about this stuff over the years. So I think it's a great question, but it is also kind of the question, our questions, and it really is what should motivate all of you to keep doing this work, because the answer is in your hands to some extent.
And let me, so I think that's spot on. And let me just add a point that this idea that, well, we'd like them to, or it'd be nice if the subjective measure correlated significantly with some biological measure, like whether it's hair cortisol or whether it's some change in epigenetic marks, whatever it might be, cytokines. I think back to one of the early studies in the hair cortisol literature, back when things seemed a little simpler than they now seem. The German group, the Kirschbaum group, did a study of long-term unemployment in Germany. So it was basically a nice simple design, two groups, matched on most things pretty well, except that one group had been unemployed for at least a year. And the other group, and they weren't starving because of various support, government support, but still unemployed, versus people who were gainfully employed over that same time period. And they measured, very simple study, they measured PSS, they did PSS and they did hair cortisol. Both measures went up significantly, or both measures were significantly higher in the unemployed group. So the unemployed group showed more perceived stress than the unemployed group, and the unemployed group showed higher hair cortisol than the employed group. So both groups went in the predicted direction, but the two measures didn't correlate significantly. So it means that, I think that's a beautiful demonstration of the fact that a particular kind of adversity or stress will have impacts on our subjective reactions, and may have similar directionality, similar impacts on our physiology, but the magnitude in each individual, the magnitude of that change or that difference may not be the same. So one person responds more with in terms of their cortisol levels, another person responds more strongly in terms of their perceived stress. That's sort of what I got out of that result. Other questions? Okay, well, as you guys know, you'll have the opportunity to provide feedback to the workshop, to us, the presenters, the organizers, about what you liked, what you didn't like. We hope you liked most of it and didn't dislike very much of it. But anyway, we value your feedback and do take it into account in future workshops. So I thank you all very, very much on behalf of myself, Richard, and the other instructors for your participation, for your attention, your great questions. And if, as I said on Monday, if you have any specific questions about the stuff that I talked about, feel free to shoot me an email and I will be sure to get back to you.
Yeah, same here.
Okay. So anyway, thanks a lot.
Thank you.